Okay, so as I mentioned, prior to connectionism launching off and sort of becoming more prominent, we were starting to learn more about the brain through a number of primary brain imaging methodologies. And so now we're going to talk about some of those. So one of them is called single cell coding, right? And so this is when you just stick an electrode or a series of electrodes into somebody's brain, right? And this, what happens is it measures the actual firing activity of neurons, so the direct firing activity of neurons. So these peaks right here, these lines, these are actually when a neuron fires. So this is like the firing rate of a neuron. And as you can imagine, sticking electrodes into somebody's brain is highly invasive and often unethical. So usually the participants who are in these studies are usually stroke patients or people who already have, uh, they already need electrodes to be put in their brain for some other reason, usually a stroke. And this is really kind of the gold standard of brain imaging methodology. So it not only does it have high spatial resolution, meaning that it can tell you, it can pinpoint exactly where in the brain the neurons are firing, so what part of the brain is firing, but it has also high temporal resolution. So when something happens, the neuron fires and it tells you exactly it fires and I should say that the peaks, these little lines that go up, directly correspond to when the neurons fire. So as a result, it has high temporal resolution. And we can contrast this with EEG, which I'm going to try to say it right. It's electroencephalography, is used to measure ERPs or event-related potentials. And event-related potentials measure reflections in the brain's electrical field generated by neural activity. And this is cheaper than other methods. In fact, there's a lot of people who use ERP and a lot of people who don't even really study the brain, right? And the benefit of this is that one of the benefits is that it's not invasive. So you just have to put like a cap with these things on your head, right? And that measures these waves, yeah, these fluctuations in the brain's electrical field, right? And so the problem with ERP, one of the downsides of ERP is that it has poor spatial resolution. So it's not clear what part of the brain generated a given wave, right? But it has high temporal resolution. And we can contrast this again with the more familiar brain imaging methodology, which is called functional magnetic resonance imaging, which is short for that is fMRI. And this actually indirectly measures brain activity. So we'll talk more about that. It doesn't actually measure the brain activity directly. It just measures things that happen when consequences of the neuron firing. It's now the downside of fMRI is it is extremely expensive in terms of time and cost. fMRI machines are extremely expensive, and so using an fMRI is going to cost a hefty amount of money. And it also takes a lot of time, right? You can only put one person in the fMRI at a time, rather than in other studies, you can run participants simultaneously. It has high spatial resolution, right? So you can pinpoint where in the brain the activity is coming from, but it has poor temporal resolution. So you can't know with certainty that a given event caused a certain amount of brain activity to go on. And then, so I want to point out the logic. So a lot of people probably have seen an fMRI scan and they think, great, what am I looking at? Maybe, right? Is that the activity itself? Is that the brain firing? And it kind of is, but it's indirectly measuring in neural activity. So I'm going to explain the logic of fMRI. So the whole idea is that neural activity, when you're using your brain and when your neurons start firing, they need oxygen. And so in order for a neuron to fire, it needs to recruit oxygenated blood. Okay, and so when a region in the brain becomes more active, cellular activity increases, which requires oxygen from the blood. So therefore, oxygen from the blood is going to flow into those neurons. And this is what's called the BOLD signal. It stands for blood oxygen level dependent signal. And that's essentially what you're measuring in an fMRI. And it essentially measures the difference between oxygenated and deoxygenated blood. So when a region in an fMRI is sort of lit up or activated, that means that the magnetic field is detecting more oxygenated blood than deoxygenated blood. And so the implication is, right, fMRI actually doesn't measure actual brain activity or neural activity. It is an indirect measure of brain activity. Okay. So it's not actually measuring the neurons firing it, but it is measuring something that neurons need in order to fire. And so one thing you can ask is, or uh, one question that researchers ask is, how does the bold signal relate to neural activity? And one hypothesis came out by Reese, Friston, and Koch. This is the Reese, Friston, Koch hypothesis, which says that the bold signal increases linearly with neural firing. So as the neurons fire more, the bold signal will get stronger. And so what they did to test this is they put monkeys, they attached these like uh, electrodes on into monkeys' brains. And what they did is that they showed a bunch of dots that were moving. They were either moving randomly with zero coherence or 50% coherence or 100% coherence. So they were either moving randomly or 50% of the dots were moving in a coherent or in the same way, or they were all moving in the same way, right? And so importantly, they put the electrode in the V5 of the visual cortex, which is sensitive to motion. So if if uh, if neural activity is related to the bold signal, right? Sorry, if neural activity is related to the bold signal, then we should find that the neurons, the firing rate of the neurons should increase as we increase the coherence of the dots moving. And and they also showed these stimuli to people in an fMRI scan, and then they correlated basically the firing rate with the fMRI scan, and they found that for every 1% increase in bold signal resulted in 9 spikes per second in the monkeys. And so that's one idea. And that study sort of suggests that the bold signal increases with neural firing. But the I can't say this, let's see, logothetis hypothesis says that the bold signal isn't actually correlated with it's not, yeah, not correlated with the neural firing. It's actually correlated to the input that a neuron receives rather than the output. So, you know, and neural activity can occur without any firing, right? So, a neuron can take in nutrients and oxygen, sorry, from the blood. And so that's the input. Input is what the neuron receives. And what we're only measuring with the electrode, with the firing rate, with the single cell coding, is when the neuron fires. And so maybe the question is, is the bold signal correlated with the input, what the neuron receives, or the output when the neuron fires? And I'm not going to, I just want you to see that this is the actual data. So to test this hypothesis, um, what I think logothetis did was put monkeys in a fMRI and then also attach electrodes to their brain in probably the V5, and then monkeys watched a rotating checkerboard, and they measured the bold signal as well as the input and output neural activity. What they found was that the bold signal was more strongly correlated with the input, which is called local field potential. You don't need to know that. I'm not going to test you on these acronyms. But the bold signal was more strongly correlated with the input than the output activity, which MUA stands for multi-unit activity, and the SDF stands for spike density function. Again, I'm not going to test you on what that is, but the important thing to take away from this is just that the bold signal was more correlated with the input that a neuron received rather than the output activity, which makes a lot of sense, right? Because yeah, it's measuring oxygenated blood flow, which is sort of the input to a neuron, not the output. So how does the bold signal relate to neural activity? It appears that the bold signal is most correlated with input activity, but output activity, of course, is also correlated with input activity. So the bold signal is also related to output activity as a result. So hopefully that helps when you see a brain scan and you'll know what you're actually looking at. You're not actually looking at the brain, the neurons firing. You're looking at the amount of oxygenated blood in that region, which is related to the input that the neuron receives, which is also correlated to its firing rate. Okay, and so the whole point of that is to show that with the advent of PET scans, fMRIs, and microelectrodes came an understanding of the brain. And these techniques often only tell us what regions of the brains are involved, but they tell us very little about how the information is processed. So rather than trying to understand how a population of neurons work, some constructed models of these neurons to see if they could explain behavior. So in the next video, we'll talk about neural communication uh, and single layer networks.